neuroplastogens is really interesting and brings some difficulties as well. And a lot of this is through the so-called psychedelics. There is a neuroplastogen you give once, maybe twice, and then all this other stuff happens, whether it's guided by therapy or maybe not, but it also poses some stuff. How do you do a dose ranging study with some of these things that has an effect for 4 or 5 months?
Well, Steve, thank you so much for taking the time to be on the show with me today.
Happy to be here.
Yeah. For the audience, Steve was the Chief Medical Officer of Karuna Therapeutics, recently purchased by Bristol Myers Squibb, having produced the first successful schizophrenia drug in decades. So I'm really excited to be having this conversation and learning more from Steve about his career to date. Please, go ahead.
Yeah, sure. And it's not so much the first in decades, but it's the first new mechanism medicine in decades. So cariprazine was out there a little while back and wasn't even a decade ago. But interestingly, things sort of happen. There was another company working on something called a TAR1. We were working in the muscarinic space, and these are all different kinds of mechanisms. And things like depression, you have a lot of different medicines with different mechanisms of action, and some work better for some people than others. And so this was a real paucity of not of medicines, but of really different kinds of medicines. And so I think this is a big step forward in the schizophrenia field to have a different mechanism. And I think there'll be others that follow pretty quickly. So.
Yeah. And I'm excited to kind of dive into your view on the landscape, but maybe just give us a thumbnail sketch of your career and how you ended up working on Karuna in the first place.
Sure. So my background, I'm trained as a psychiatrist, so a medical doctor, then residency in psychiatry. And then I did a research fellowship, and it involved neuroimaging. And that's been sort of foundational to my career. We had this radical idea that the brain was made of circuits. And of course, that's the normal thing that we think about now. But so I've sort of said, if you have something and it involves a circuit in the brain, I had half a chance of understanding it. Anyway, I was in academic medicine for a while, did neuroimaging research, mainly with mood and anxiety disorders. And then Eli Lilly came and made me an offer I couldn't refuse. So I went and worked on an antidepressant, duloxetine or Cymbalta there. And then I've been with several companies since then, both large and small. Been fortunate enough to be involved in a number of different things that actually got approved and on the market. And then the last one really had to do with Karuna Therapeutics. So I joined 2017. They were sort of reviving it and had this great new idea. And so I was employee number 2. And so I saw the whole thing really from almost the start to the end. So that was sort of exciting. You usually only get to see a part of the story, not the major part of it. So, and we were very fortunate to we never skipped a beat. We never really had a stumble. So things went very quickly. And then recently, of course, it was approved and is out on the market as of, last fall. So,
yeah. Well, congratulations on the incredible milestone there, Steve.
Thanks.
And kind of prep for this conversation, I was reflecting back on an episode I did with Dr. Anantha Shekhar over at UPMC.
Oh, yeah.
And he mentioned that the compound was essentially going to be dropped by Lilly as an Alzheimer's drug before you spun it out. I'd love to get the kind of inside story as to how you identified this as a thing that needed spinning out in the first place.
Well, and others were more involved in that than I was, but Anantha and then Alan Breyer and even Steve Paul, who come back into the picture later, at Lilly had done this, and it was interesting. This molecule looked like it was they were using it for Alzheimer's, and then they sort of serendipitously found that it was really good for what they were calling behavioral problems associated with Alzheimer's, but it was really psychotic symptoms. And it looked for all the world like this drug was really good at that, but it also had a number of side effects, and they weren't able to kind of figure out how to make it more tolerable. And so it did sit on this shelf for a long, long time. And then Karuna, in, I guess, 2010 to 2017, sometime in that area, they sort of looked at this, talked to some people, and they said, this was really good. And Anantha had done this study in schizophrenia patients, real small, 10 and 10, but it showed this huge effect. So there were people out there saying, if you can just harness this, this would be a great drug. And what Andrew Miller and Eric Alinko and some of these other people did, they were at what we call an incubator. So in Boston, there's this incubators like Third Rock or PureTech, and they have a number of different companies. And so they sort of help them all grow. And then if one of them makes it, they can do another 10, and two of them
Sure.
make it, they all do really well. So and we were one of the ones that did really well. So they and the industry had sort of been looking at this muscarinic issue, and a number of the companies I had been working on, we were all looking at it, but in the wrong way, and sort of what we were trying to make it more sensitive to one type of receptor than another, which is a not uncommon thing that people do. But what Karuna did is they sort of said, well, most of the benefits are central in your central nervous system, and most of the side effects are peripheral. Now, your central nervous system is protected, so not all things can get in there. So what if we block the peripheral side effects with an anticholinergic? The muscarinic is procholinergic. And but if the anticholinergic can't get into the central nervous system, then we'll preserve the benefits and mitigate the side effects. And that essentially is exactly what happened. So when I sort of heard about this, when they were talking to me, I was like, wow, why didn't I think of that? That's so simple, but it's this Gestalt-like thing, and once it's out there, then people are like, oh, okay, yeah, that makes sense, and so on and so forth. So anyway,
I'd love to kind of dive into that a little bit more. What, you were moving at the time from Eli Lilly to go join Karuna, is that right?
No,
Okay.
that was, I went a long career from Eli Lilly to Cyberonics to Novartis to Takeda. I had been at Forum, which was a company looking at cognition and schizophrenia. And so they had unfortunately not done well in their phase 3, and so the company dissolved. And so I was available to then be recruited at Karuna.
Yeah.
And yeah, go ahead.
Well, I was going to say, and having seen, it sounds like the largest of large pharma, the biotech adventure, how did you think about making the switches back and forth? I don't think I've spoken to a lot of folks who've gone from one and the other, and then back and forth in the same way that you have.
Yeah, I this may sound a bit arrogant, so I apologize, but not everybody can do well at both small and large. And there's some, you get into, and there's pluses and minuses to both. At Takeda, before I went to Forum, I was the head of late clinical development, very large company, and you had a lot of resources and you could do a lot of things. But you also had a lot of layers and a lot of people who were wanting to contribute to your studies, and that's not always useful, particularly in CNS. So the smaller companies are a little more agile and innovative. They can take risks that the big companies don't. Like most big entities or bureaucracies, big companies tend to not be the risk takers. And so this development program was very risky, so this was a new mechanism. People who fund this stuff like, oh, is this the same mechanism? Because then I can fund it. If it's a new mechanism, I'm not so sure. So that's always a barrier. And then this whole idea of combining the antagonist with the agonist was, different and new. And so that boggles a lot of people. And then once we were able to get on track and get going, we were able to move quicker than many of the larger companies do. But we won't go into that. That'll be another discussion sometime down the line.
I was going to follow up and ask about that, but yeah, please. Yeah. I mean, I was going to say, as you kind of mentioned, you almost experienced this like charmed path
Yes.
along the way. Like,
Yeah,
what do you attribute that to?
It was sort of the right time and the right people. So when I joined, Sharon Sawchak, who is our ClinOps person, what came right with me, although she didn't join the company. She was more of a consultant for a while. But we had a lot of experience. She'd been at GSK for a long time. I had this career path and had been at very high levels of stuff. So I strategically knew the whole pathway from early to late. And then I knew a lot of the CROs and stuff like that. So I could just, and sites, a lot of the sites that were available knew who I was. I sort of tweaked some of the early Karuna people by saying, yeah, the sites that are working for us, they're not doing it because they know Karuna. That means nothing to them. They're doing it because they know me and they know Sharon. So we knew the right sites. We knew some of the CROs. In talking to some of the CROs, and just like myself, one of the reasons I went to Karuna is I was looking at this. I'm like, wow, that science is really cool. That's great. And if it works, this is going to be wonderful. And everybody likes this. So when we were going out there and I would tell people, they're like, wow, that's really cool. And so we got a lot of help from some of these vendors like Syneos and Signet that maybe not all small companies get, partly because they knew who I was and my track record, as well as Sharon. So we were both very well-known entities with good records. And we were working on something really cool. So that combination really got us a lot of high-quality resource and attention that small companies don't always get, in my opinion.
Yeah. How much of this is your ability to take the science and tell an incredibly inspiring story?
That is really key. And this goes way back to when I was in neuroimaging. I worked with somebody named Helen Maber, who's now really well known, but she was just starting out. And when you do the neuroimaging, it's like you have all these blobs, and we would color them. So they call it blobology somewhat derisively. But you're looking at this and what does this mean? And where she was really great is she said, no. Here, this is the amygdala. Remember, it connects here, here, and here. It connects to the hippocampus. You can see it here. And being able to concisely and clearly delineate a story out of the somewhat chaotic mass of stuff that's going on is really a key. And I think that did help. And when Steve Paul joined us a little bit later, he was really a masterful storyteller and could really, especially with the money people, he is like, look, here's why you need to invest and so on and so forth. And he was really great at that. So we had some really good people. And as we grew, I think we also hired above average folks, going along with us for the most part, not all the time. So that also helped. The other thing I talk about that's been happening like maybe 20 years ago, the CNS trials really were hitting the skids. Not much was working. There's still problems there. There's all sorts of problems we can talk about in a little bit, having to do with placebo and precision of diagnosis and all these sorts of things. And one of the things I talk about a lot in this clinical trial methodology is you learn from your mistakes and from other people's mistakes. And I've been round enough, you can see my gray hair, so lots of mistakes. A lot of my own, some of other people's.
Sure.
And with Karuna, we just didn't make the same mistakes. So we knew what to avoid. We knew the pitfalls and really avoided pretty much everything. So
say more about that. What were the mistakes that you intentionally designed around?
Well, one thing, I'll kind of put these in phrases, but more is better. No, it's not. When you're doing these trials, you're trying to see a signal. And there's some difficulties with, especially psychiatry, but CNS in particular, that a lot of our endpoints are soft. So unlike a hemoglobin A1C or doing a neuroimage of a tumor and see if it's grown, you don't have that. So depression, are you feeling less depressed today? Yes or no, or less anxious? It's not a very hard, solid thing. And by the way, it fluctuates during the day. So you have to deal with things with what I call a lot of variance. So you're looking for a signal and you want a large signal. And fortunately, the Cobenfy, the KarXT that we have had a big signal, but you're also trying to diminish the variance, the inherent variance that is occurring in these trials. And so there's a number of things that you can do. Now, there's probably a limited number of really high quality sites doing this kind of work in the US. And so the problem with doing too much is like you go past, I won't say they're bad sites, but you go past the A sites into the B sites and then the C sites and then the D sites. And so you just add more variance and it makes it more difficult. The other thing is to go at the right pace. Now, I have nothing against going fast. I like going fast, but when I have a car in front of a brick wall, I don't accelerate. It's just,
Sure.
I used to ask people in talking to those folks is how many times have you been asked to make a trial go faster? And everybody holds up their hands. I ask, how many have you been asked to slow down a trial? Nobody holds up their hands. And so this huge imbalance, just tells you something. And people sort of can't help themselves. When I was at Takeda, we were looking at an antidepressant, and we had the first wave of studies in the US, which was 0 for 8. The second wave of studies was 4 for 5 positive. So 0 for 8, 4 for 5, same compound. What was, well, one of the things was, not getting in a hurry and doing stupid stuff. And when we were making that transition, I remember talking to our staff, both Lundbeck and Takeda, because we were doing it together, and said, okay, I'll guarantee you one thing about this second wave of studies, they will not be as fast as the first time. Does everybody understand? Yes, we understand. Okay, fine. Thanks. Great. So that means you're not going to come and say anything. No, we're not going to do that. I'm like, great, I'm from Texas. See this scar on my thumb? Get out a knife, cut your finger, and sign in blood. Ha ha. No, we're not going to do that. Pause. How long was it until somebody asked me to go faster?
Let's go 1 month.
Oh, it was actually it was 6 months.
6 months. Okay. All right.
But yeah, it was both Lundbeck and Takeda, their upper management, not talking to each other, but within a week of each other, they were asking. So it's just inherent. They can't help themselves. And you need somebody, that has the courage to sort of say, no, we're not doing that. Here's the reasons. You just can't say no, but remember why. Go ahead.
We had diminished variance. We had right pace. Was there anything else that was critical here?
Yeah, you need a collaborative stance with your sites. The sponsors run these trials. You have these big CROs that are helping you kind of do this. And then there's this site networks all across the globe. In this case, we were just working with the US. And having had experience with them, and they had experience with me and with Sharon, which was the ClinOps team is really key in working with these sites. And so you have to be able to work together sort of hand in glove. And so you're never completely on the same page. So, but you have to go to them and say, I need the right patients at the right sites. Sounds simple, but it's a lot harder. And I think you're one of the right sites. We're going to work with you. We're going to watch everything. But, I want to work with you and give you what you need to get the right subjects. And, that we have inclusion and exclusion criteria, but that alone doesn't quite get it. And a lot of these people, the PIs in particular, will have this sort of sixth sense. Somebody comes in, they're like, they're not quite right. I can't figure it out. And what I want them to feel is I don't jump in their face, but I want them to feel my hand on their shoulder at that moment and say, ooh, I wonder if Dr. Brannan would really want this patient in the study. And again, you have to work with them. We had somebody in one of our Karuna studies, we had the PANS from 80 to 120. So 80 was the cutoff, and anybody scoring an 80 on the PANS probably needs to be hospitalized. So 120 is really way out there, and they brought in somebody at a 118 or 119 at screening, and then there's a time before they get to baseline. And I remember talking to him, I said, I don't think you're going to make it because this person's already, really psychotic, and you can't do the study as well. You just treat them. And they're like, no, I think I can do it. And I was like, okay, but if they're above blah, blah, blah at the baseline, then I'll try not to say I told you so, but, and of course that's what happened. And so they came back and they were like, I thank you for letting me try. I don't know what I was thinking, but I promise you I will get you the right patients as we're going forward. And at that moment I knew, that site was going to do well for us, which they did. So in hindsight, go ahead.
Yeah. In this dynamic where, there's a handful of sites that, are incredibly high quality that you want to work with, how do you think about competition at the sites? You are the only one working on muscarinic. Yeah.
Well, and when we first started, by the way, there was only one other company running inpatient schizophrenia trials, and that only overlapped by a few months. So that was also timing. That helped us. There's no doubt that when there's less competition, you're going. And now, after we've succeeded, now there's a bunch of people competing for these subjects, so there'll be inherently more variance, and the quality will suffer a little bit. I think one of the things, and again, I don't know why people don't do this because I don't think it's rocket science, but apparently it is. It's like when we put together a protocol, and I'm like, okay, this is something I want to do. So I'll send it to the CRO. I'll send it to a couple of sites. And it's not that I think any one site knows more than I do about a trial, particularly a specific trial, but no one person knows everything and sees everything. So every time I've done this, somebody has come back with not just one, but usually, a handful of things, if not more, of like, oh, this would make more sense if you did this or this. And for the sites, like they're looking at, if I bring in a patient on this day, if you've got 5 different things, I can do this, but if you got 10, how is this all going to fit together in a reasonable way? So they can be very, very helpful in those sorts of things. So, and just the fact, since people don't do this, so we do. We ask for feedback. We don't take all the feedback, but we listen to everything, and I think they know that. So that's part of it. And then some of the sponsors, and some of these are small biotechs. They're small. They don't have enough people. They don't know better. They might just leave it to the CRO to run the sites. We don't do that. Like, talk to me anytime, day or night. So. And I wouldn't, especially toward the end, I wasn't the person they would necessarily call first, but having that availability. And then there are certain meetings throughout the year that, being a small company, we couldn't go out and visit people. We didn't have the money or the time. But we could, these meetings where people go to, like CNS Summit or stuff like that, or ASCP, we always said, we're here at the meeting, we want to talk to you. And so we would spend time talking to the sites or the site networks extensively. And
Certainly.
I would bring my ClinOps team. So,
Certainly.
Anyway.
I think that a common thread that I've seen in some of the smoothest programs that I've witnessed has been a direct relationship between the sponsor team and the site team to build that trust, to build that collaborative experience where you really do listen to feedback and iterate on the basis of it.
So BMS came with us to the ASCP meeting. It was sort of right after the transition had occurred in mid-March. This is sort of into May. And so we were with one of these site groups, and at the, I'd said something about, don't talk to me, talk to these people, because I'm just a consultant now. And so dessert they brought out with, black candles and stuff like that. Like, so it's for my demise or whatnot. And they all sang a song and whatnot. And so I'm walking back with the BMS people, and they're looking at me and they're like, they really like you. I'm like, yeah. It's like, I never thought about that. I'm like, well, it's important. They're like, why is it important? I'm like, because there's 3 other trials out there, competing for the same time and space. And I don't think people, well, I'm sure my competitors might disagree. I don't think people gave us more than they should have, but they certainly gave us a fair shot in the grouping of stuff. And then you also have to watch things. So we have something called blinded data analytics. And so there was a site that had 3 people over the age of 55. The median age in the trial is like 42, 44, something like that. So it's certainly possible, but it's highly unlikely. Not 3 in a row. 1, if you don't even pay attention. 2, you sort of look at it, and then by the time the 3rd is like, So I called up the site, but you don't jump down their throat. I just called the PI up, and I said, hey, how long is the geriatric convention in town? He's like, what are you talking about? I'm like, well, I saw your first 3 patients were all over the age of 55, so I couldn't figure out why that would happen. And then I thought, oh, there's a geriatric convention in town. How long is it in town? He was like, okay, I'll get back to you. And he never answered the question, but the next 3 patients they did were all under the age of 55. So, and I know what he was doing is there was another trial from one of our competitors that had an endpoint of 55. And so they were giving us the ones over that age, and then all the others were going into the other trial. And I was sort of subtly reminding him that didn't particularly like that. So,
Right. Savvy.
well, that's the advantage of years, right? Experience.
Well, I think that kind of gentle touch is important to kind of reflect on, because it's a way to kind of stay aligned without antagonizing.
You got it. That's exactly right. You can't be a mean bastard son of a bitch, right? People don't like working with you that way. But you can get your points across. I use humor a lot to do some of that, and that seems to work for me. But
Yeah, certainly. Oh, do you think that this, this gentle touch is a thing that's easy to propagate at a BMS? Like,
I don't think it's easy to propagate anywhere.
fair.
And I think, yeah, I've even had some people talk to me. It's like, can you write a book or do a videotape or a podcast or something,
Right.
to
This will be
help
it. This will be the canonical podcast.
okay, so you'll be the start.
Well, you'll be the star. I'm the interlocutor.
Oh, sorry, I meant start.
Oh, the start, of course.
Yeah, the beginning. In the big companies in particular, you can get people who just make a living saying no. And when I hire people, I look at this, and it's like, what are they really in this for? Are they just in for themselves, or are they doing it for the good of mankind? There's usually, as a psychiatrist, there's mixed motivations. There's always mixed motivations, but I do look in that mixture. I look for something other than just themselves. And oftentimes when you have that, for the most part, there can be, particularly for clinicians, people can be pretty direct. And I had to learn over the years to be a little less direct and a little more diplomatic. But I think people's desire to sort of collaborate and work as a group is an important thing in these sorts of things. So if you can get that and sort of accentuate the teaching in that direction, that's probably how you need to go.
Certainly. Hey, I would love to just backtrack very quickly to a discussion we were having around the right pace.
Yeah.
How do you know when you're on the right pace, when you're going too fast, or if you're actually taking the time you need?
Yeah. So there's a couple things. One is, and we would do, as a small company, we didn't have the internal capacity, but we would be talking to several what we call CROs, or contract research organizations. And so we'd say, and then with experience, like Sharon would know this off the back of her hand, but what is typical for a site to do? And so it's a half a patient per month. And then, so that's 6 per year, blah, blah, blah. And so you calculate your timelines. And then I've learned, because people get too literal about this, is say, oh, the timeline is going to be 12 to 18 months or 15 months plus or minus 3. And so they seem to be able to handle that a little bit better, because if I tell them it's going to be 15 months, then they're going to just, at 15 months, they're going to be like, oh, it's 15 months. Yeah, something wrong. And so and again, this is a range, and you can look historically at ranges, particularly CROs can with particular sites. And then you look very closely early on, and this was in the second wave of studies we did at Takeda with Lundbeck. And Lundbeck, quite frankly, I think was the ones who did this. When sites first come in, you let them kind of go, but you watch very closely. So you watch the quality of the data. You look for what we call deviations, or very from the typical thing they're supposed to do. You look for any signs of quality problems. You might look at of the patients that come in. I told you there was this 80 to 120. You usually don't have to worry about the high end, but if they're supposed to be at least 80, you don't want a bunch of patients, 80, 81, 82, 85. You want to see a spread, because that's the normal way things happen. So in our trial, the mean was like 97. And for most of the sites, you saw this normal distribution. So you're looking for things like that to make sure things are not amiss, or there's no red flags or discrepancies in what you're doing. So And then there's some things now they do with raters. So a lot of this we have to do, again, these are soft measures, outcome measures. And so like the PANS is this questionnaire you basically have, and you need trained raters. And then you have backups to the raters. You have the site raters, which are the better raters in the most part, but you have central raters to sort of back up, and they're rating the same audio tape or video audio tape. And, if the site rater has something, a couple points lower and the central rater has it a couple points higher, it's like, then you look at all these things. And so you keep watch on this constantly. So, and then there's a little bit of a feel. So when you're first starting out, things are a little bit clunky, and then you sort of see that things are sort of like moving on a regular pace, but not too fast. So we had one site that, the very first week they had 5 patients come in. Now, these are acutely psychotic patients. So mostly, I don't care where you are in the world, you're not going to have 5 acutely psychotic patients come to you in the same day, certainly, much less the same week. So it's like, what's going on here? So
it sounds like a key to all of this is looking at the stream of data, looking at deviations, and trying to pick out any funny business.
yes, I'd say that's a good part of it. Yes.
Maybe that's the title of the book, the inevitable book. Looking for funny business.
Okay.
Hey, I want to stay on this subject of soft endpoints. We've had Ray Sanchez from Cerevel on the show as well, and we kind of asked him what would he like to kind of ask you. And one of the questions that came up with that kind of came up in the prep was talking about the trajectory of precision psychiatry and how it might evolve in the kind of landscape of psychiatric trials, which have historically have these subjective endpoints.
Well, we certainly need it. I mean, and it's not just in terms of the outcome measures, it's also diagnosis. So somebody walks in, and a good psychiatrist should be able to distinguish certain things, but like there's things coming in, like somebody who's bipolar but psychotic, or schizophrenia and psychotic. It's really hard to tell the difference between them, in a couple of days or even a week. So we don't have something like, if you're in diabetes trials, you had a hemoglobin A1C. So that is hard to fake. And so you have a much more precision in terms of knowing stuff. So it would be great if we could start, and people have been talking about this and probably promising too much too quickly, frankly. But I think, this is also part of the future as well as the present, but these so-called digital biomarkers. So in schizophrenia, we've known for over 100 years their speech is different. But now we can record, just like you're recording me now, and play the stuff back and look at all sorts of things that we normally don't hear in terms of tone and pauses and all this kind of stuff. And you can track it with the AI and similar sorts of things. And you see a pretty big difference. So these are things I think are going to evolve in the next 5 to 10 years. And I think they're sorely needed, because even things, I talk about schizophrenia or depression, they're not one thing. They're probably 3 or 4, maybe more kinds of things that we all bunch together. And so if we can disentangle that, I think our treatments will get better and these trials will get better. So we'll know that we've got, the right, more assurance that we have the right subject in for that trial.
What do you think it's going to take to bring these digital biomarkers through to research and clinical practice?
Yeah. Well, and it may come to clinical practice before it does the research. One of the big needs we have is for both sponsors and the regulators to move quicker than molasses. So it's not just the regulatory folks, because the big companies in particular can be very slow to adopt something. So in one of our trials, we put something in. We didn't put it in the pivotal trials, but it was in the safety trials, so something called ecological momentary assessment. So this is a very new thing in the last 5-10 years. And maybe it's longer, but at least in terms of, being up in front of people. And it's basically, you've got these little phones, and so they can send you out a text or a message or whatever. once, twice, 3 times a day, maybe more, and say, oh, where are you? Who are you with? What are you doing? And you get this sort of information, but also are the voices bothering you? And so we stuck this in some of our safety study, and it's amazing what this looks like. And you have a lot more power because you're not just asking somebody once a week at that's about as quick as you'll do stuff with people. And over the last week, how bad were the voices? And it's hard for some of us to remember exactly what we ate for breakfast 3 days ago, much less other stuff. So
Yeah.
I think there's going to be a lot more power in this, and I'm hoping that there'll be enough flexibility without being crazy about stuff. But let's figure this out. Let's use this because we really sorely need it, and I think it could really help a lot of these trials. There are also going to jump in on this what are called duplicate patients in these trials, and this is also in more tech savvy places. So there's both pluses and minuses to tech, and these are people who are faking schizophrenia or psychosis or faking depression or whatever. It's really fraud. And I don't have a warm, fuzzy place in my heart for these people. For the most part, they're just committing fraud. But nonetheless, we have to do all these things to try and keep that to a minimum. And I think one of our successes is we really did keep that to a minimum in our trials. Unfortunately, a lot of people are not doing this. So as I've said, it's sort of like, a beware of dog sign for your burglars in your backyard. It's like, oh, there's another house I can go to that doesn't have a dog that I can rob.
Yeah.
So.
Well, certainly we've been seeing a trend towards more automated systems for checking for potentially fraudulent entries in research studies. Yeah.
That first wave of studies at Takeda, as I told you about, in the very first 2 studies we looked at, there were 23 people in both studies at the same time.
Wow. Yeah.
So it is unfortunately a necessary evil. It's sort of like all the TSA stuff you have to do at the airport, right? I didn't have to do in my youth, but you have to now.
Well, back to this theme of monitoring the data and looking for funny business. This is just another form of funny business. Hey, one of the things that comes up as we explore vocal biomarkers, it's a thing that we've explored on our end, whereby when patients sign up on our platform, we might have a conversation with them, and then kind of use that to kind of inform some of these vocal biomarkers. One of the things that has come up is there a downstream impact on the regulatory submission that is required if that's a kind of criteria for entry into the study?
That's a tricky question, and there's an implication for it. But they are okay with you enriching your trials. So, and there's a whole guidance on this. And from my understanding of that and my understanding of vocal or facial biomarkers, that would be fine. Now, you might get a line in your label that says, that this drug worked in people who had met these criteria and met the vocal facial biomarker, blah, 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 blah. So you might have a little bit of a limitation there, but that wouldn't be all bad either, because then whoever, then you could start getting into clinical practice like, oh, maybe you want to do your vocal biomarker, which is pretty easy. Should be able to be done, I think, in the future fairly cheaply as well. So
I guess somebody needs to start with a validated diagnostic.
Yeah, it's the validation piece, which can take a long time. But I don't think you need to have that all fully validated for the enrichment. So, for instance, we don't take on, the PANS between 80 and 120, so that's in the label of what we did, but the FDA label doesn't restrict it to people who are not between 80 and 120. So,
certainly. One of the things that has been a theme on this show and some of the guests we've spoken to is this idea of us being in a neuroscience renaissance, if you will. I'm curious, what do you attribute that to?
These things always are cyclic. They go up and they go down, and some of it doesn't make any sense. But one of the things people ask is like, well, why didn't Lilly do what you guys did with xanomeline, 30 years ago? And the answer was they couldn't have. They didn't have the techniques. They didn't have the things that could be done today. They tried sorts of different things, but the field had not advanced. And the field is just advancing so quickly on so many fronts. We have a lot better genetics. We have a lot better imaging, just all sorts of things, just even techniques, clinical methodology. We have caught up to some of these, duplicate patients. Non-adherence is another big issue, especially like in schizophrenia trials.
Sure. Yeah.
Actually, sorry, this is a bit of a non sequitur, but we've been talking about face and voice. So one of the things that typical schizophrenia patients do not have is good social cognition. And so we were playing back on one of our subjects, this patient, and he was telling a joke on the tape. And according to the person who listened to it, it was a pretty good joke. And then he stopped and thought, wait a second, how many acutely psychotic patients with schizophrenia can tell a joke? The answer is 0. So, there's things you can pick up. But I think in the future will be a lot more subtle and more precise.
Yeah. Another question from Ray. Are you optimistic or pessimistic as far as AI and defining clinical development and its applications?
In between, I'm
neutral.
Well, people used to ask me, in high school, like, what's your favorite color? I don't have a favorite color, but my shirt here is black. So I used to tell them black because I learned if I said black, they'd stop and leave me alone.
Right.
But it wasn't my favorite color. AI is going to be, even this year, useful for certain things. Part of the problem is people come in with all this hype and it's like, oh, this is the best thing since sliced bread. By next year, everything will be run by AI. And it's like, no. And when they say they can figure out new targets, no, they can't, because no one knows what the new targets are. So just don't go there. But there are things that it can do very quickly. It can monitor, the interviews that we do for quality and for all sorts of things, just very quick. It never gets tired. It doesn't get grumpy.
It's consistent.
Well, in the standard acute psychosis in schizophrenia trials, there's a bunch of them out there, and you could probably get AI to do this. Now, don't make it your final draft. Don't be stupid. But let it do that. It could do a clinical, the CSR, the clinical scientific report of the study. It could take a protocol, and then we have our CRFs, so our report forms that we have to do, and it can do all these things. Again, first, maybe second round, not your
draft.
Final. Yeah, draft, and this saves time, so it'll be good. So there are those sorts of things, just as long as you don't. Now, some of the more subtle things, we're talking about, like how do you interact with your site with that soft touch on the shoulder? Can't get there. Can't do that. Or this feel about something's not quite right with this patient. Probably can't do that. But it will, if you pair it with some of these digital biomarkers, if you or I were to look at all the stuff and do the measurements, we could do it, but it would just take us a couple of days, and it's going to take 2 minutes. So you can get back to the sites in a very timely manner on the digital biomarker, for instance. So.
Yeah, certainly. I think that analyzing call recordings, these kinds of things, incredible opportunity, drafting documentation based on whatever's in the information, incredible. You did mention pessimism around target design, and you kicked off this conversation with excitement around targets. So I'd love to just hear you opine on what you're seeing and what you're excited about.
Yeah, so the neuroplastogens is the really interesting. It brings some difficulties as well, and a lot of this is through the so-called psychedelics. So again, talk about something that was overhyped. I'm old enough to remember some of the psychedelic stuff and walking through Harvard Square and looking at people who'd overdone it. Kept me from trying any of that stuff. But there is a neuroplastogen is something you give it once, maybe twice, and then all this other stuff happens. Whether it's guided by therapy or maybe not. But it also poses some stuff. So we talk about dose ranging studies. How do you do a dose ranging study with some of these things that you give, and then it has an effect for 4 or 5 months? It's going to be pretty hard to do that dose finding study. So there's all sorts of pluses and minuses that go with this, but we're just scratching the surface on this. Do you have to have a hallucinogenic experience or not? I'm not sure you do. So, and they're talking about micro doses again. So we're always talking in the dosing about enough to have an impact, but not too much to overdo it, and maybe throw something else in your body off balance, so to speak. So I think that's really interesting. And then I already told you about this ecological momentary assessment, which is part of this whole new range. And there's all sorts of digital biomarkers. There's stuff you can do in Parkinson's disease for just movement. So we're on the brink of a lot of things, I think, that could be very powerful in terms of giving us better insight or more granular data on what's going on. So, and then just the field itself is also progressing at a rapid pace in terms of some of the imaging stuff, genetics. We're a lot, brain's very complicated, and genetics is very complicated. So you're not going to have this huge jump, but you're going to have better and better granularity built out over the next decade or so, in my opinion.
There's this implied thesis that I'd like to name here for a moment, which is that precision, like precision medicine in psychiatry, doesn't necessarily need to be revolved around biomarkers in the traditional sense, right? Genetic biomarkers in the traditional sense, as we think about it. I think there are interesting digital precision components in psychiatry that, to your point, can use video, can use audio, can use video recordings of movement to kind of inform some of the decision making.
You can just use movement devices. It doesn't have to be video. So how people move, how they did some of this on the schizophrenia trial, long-term schizophrenia trial, and so people would be like, where are you? Well, I'm home alone. And then the actigraphy would basically not show a lot of movement. But if they said, oh, yeah, I'm out and doing the then the actigraphy show, so this gets back to your side, watching for things and making sure there's no funny business. But you can tell a lot even just from actigraphy. And sleep is a whole nother thing that they're working on. I think they're going to have some devices that'll give you some really good information without having to go into a sleep lab, which is expensive and problematic for a lot of people.
Yeah. And talk about patient burden, right?
And decrease patient burden, which Lord knows is the patients or the participants can tell you in these trials can be a real pain in the butt.
Absolutely. Maybe kind of the last kind of topic I'd like to linger on here very quickly. You mentioned neuroplastogens as an interesting topic of kind of exploration here. It seems like the kind of bulk of the discussion now is whether they will resonate in a regulatory kind of landscape. I'd love to hear how you're thinking about that today.
Well, and some of the stuff I mentioned makes it difficult for the regulators. So it's like, what is the dose range? Not only do we not know for this particular compound, we're not even sure how to do it. So that just makes it hard to write the label and stuff like that. So they do have some legitimate concerns about some of this. And then some of the people in this field, like, I'm probably going to get this wrong, but we don't need to do tox studies because, enough people took this in the 60s and 70s that we know it's safe. No. Safety is still important. Or, they got high, but we didn't put that down, as a potential side effect. It's like, that was stupid. So, learn a little bit about what's going on. And so, some of the Lycos, which was the first one to go up, they just didn't really know exactly how you chat with the FDA. I guess that's another thing you learn with experience. There's ways to approach it and ways not to approach it.
Are you keen to get in the game and apply your kind of history of expertise to this field?
Not exactly. Let me phrase it slightly differently. So there appear to be a number of companies, including in this field, that would like me to assist them, in some of the stuff. And again, I'm happy to assist people on things that I think, like, we were talking about earlier. If there's things that can make these better studies and get things or determine whether it's good or not for patients and do that more quickly, more efficiently, I'm happy to do that. So, and right now, because of all the things that are going on, and there's stuff like PTSD or post-traumatic stress disorder that really we don't have anything that effective for. And if this stuff can be effective, at least for a subset of them, yeah, I'd love to see that happen. So,
well, you heard it here first, folks. For the psychedelic companies who follow along, Steve is open for consultations.
Oh no, please don't. I have too much to do right now. Ray Sanchez. Talk to Ray Sanchez.
Ray is also open for consultations. Steve, I've so enjoyed this time. My last question to you here is, what do you not get asked enough that you wish you were asked more?
Well, when people, talk about our success and whatnot, it's the clinical trial execution. People, this is a couple years ago, but I remember there's a big investor meeting in Boston, and they were talking about all these things, and they're what they look at, blah blah blah. And I said, "Well, do you ever look at the clinical operations team?" And they're like, "No, why would we do that?" And I'm like, "Because the execution of the trial is like one of the major differences of whether you succeed or not, at least in CNS." And they sort of nodded their head, looks at me, and then went on to their conversation. It's
a little bit of the unsung heroes. A little bit of the unsung heroes, huh?
Yeah, exactly. And I think the fact that it's not asked enough is partly because it's not emphasized enough. And some of the things that you and I talked about in terms of the speed, optimal speed, and blah blah blah. There's things out there we know, but people just blow them off or think that they can ignore that side of the business. But clinical trial methodology is the key to really having consistently good trials going and executing.
Well, I'm so glad you ended it there, because it is a foreshadow for the next kind of season that we are pursuing. So stay tuned as we kind of a deep dive on ClinOps excellence.
All right. Thanks, Brandon.
Well, Stephen, thank you so much for your time.